celebrate not only the enduring legacy of two exceptional Danish scientists, but also the spirit of inquiry and intellectual pursuit that defines our academic community. Professor Neergaard and Professor Smedegaard's affiliation with the University of Mysore has not only enriched the Department of Botany, but also fortified the Department of Applied Botany and Biotechnology, propelling both the departments to new heights of excellence. ಬಂಧುಗಳೇ, ಕಾರ್ಯಕ್ರಮವನ್ನು ಪ್ರಾರ್ಥನೆಯೊಂದಿಗೆ ಪ್ರಾರಂಭಿಸ್ತಾ ಇದ್ದೀವಿ. ಮಹದೇಶ್ವರರನ್ನ ಕುರಿತ ಭಕ್ತಿಗೀತೆಯನ್ನು ಈಗ ಪ್ರಸ್ತುತಪಡಿಸುತ್ತಿದ್ದಾರೆ ಡಾಕ್ಟರ್ ನಿಂಗರಾಜು ಮತ್ತು ತಂಡದವರು.
ಹೂವ ಚೆಲ್ಲಿರಿ ಮದೇವ್ಗೆ ಹೂವ ಚೆಲ್ಲಿರಿ, ಹೂವ ಚೆಲ್ಲಿರಿ ಮದೇವ್ಗೆ ಹೂವ ಚೆಲ್ಲಿರಿ. ಆ ಆ ಆ ಮಲೆಯ ಸ್ವಾಮಿ ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಏನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಏನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. 77 ಮಲೆಯ ಸುರುಳಿಸುತ್ತಿ ನನ್ನೊಡೆಯ, 77 ಮಲೆಯ ಸುರುಳಿಸುತ್ತಿ ನನ್ನೊಡೆಯ. 7 ಬಾಯಿ ಒಂದುತ್ತದ ಒಳಗೆ ಯೋಗಿಯಾಗಿ ಒಳಗವರೇ. ಆನೆ ಮಲೆಯ ಸ್ವಾಮಿ ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಜೇನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. ಕೊರಳಲಿನಾಗ ಸರ್ಪವ ಹಿಡಿದು ಸುತ್ತಿಕೊಂಡವರೇ, 16 ಅಂಕಣ ಗುಡಿಯ ಒಳಗೆ ನಗುತ್ತಾ ಕುಳಿತವರೇ. ಮುತ್ತಿನ ಜೋಳಿಗೆ ಮುಂಗೈಯಲ್ಲಿ ಧರಿಸಿಕೊಂಡವರೇ, ಎಪ್ಪುರಿಗೇರಿ ಎಜೇನು ಮಲಗೆ ಒಬ್ಬರಗೊಂಡವರೇ. ಸತ್ಯವಂತರ ಸತ್ಯವ ನೋಡುತ್ತ ಸಾಲೂರು ಮಠದಲ್ಲಿ ಕುಳಿತವರೇ. ಆನೆ ಮಲೆಯ ಸ್ವಾಮಿ ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಜೇನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. ಆ ಆ ಆ ಬೇಡುಗಂಪನರ ಬೇಡರ ಕಣ್ಣೈನ ಭಕ್ತಿಗ ಒಲಿದವರೇ, ಮಲೆಯ ಸೋಲಿಗರ ಮನದೇವರಾಗಿ ಮನದಲ್ಲಿ ತುಂಬವರೇ. ನಾಗುಮಲೆಯಲ್ಲಿ ನಾಗರ ಎಡೆಯ ನೆರಳಲ್ಲಿ ಮಲಗವರೇ, ಸಂಪಿಗೆ ಹೂವಿನ ಸೊಂಪನು ತಮ್ಮ ಪರುಷಗ ಗೆಲ್ಲವರೇ. ಬೆಟ್ಟಕ್ಕೆ ಬರುವ ವರ್ಷಕ್ಕೆ ಸ್ವಾಮಿ ಮತ್ತಷ್ಟು ಭಾಗ್ಯ ಕೊಡುವೆ ಎಂದು ಆನೆ ಮಲೆಯ ಸ್ವಾಮಿ ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಜೇನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. 77 ಮಲೆಯ ಸುರುಳಿಸುತ್ತಿ ನನ್ನೊಡೆಯ, 77 ಮಲೆಯ ಸುರುಳಿಸುತ್ತಿ ನನ್ನೊಡೆಯ. 7 ಬಾಯಿ ಒಂದುತ್ತದ ಒಳಗೆ ಯೋಗಿಯಾಗಿ ಒಳಗವರೇ. ಆನೆ ಮಲೆಯ ಸ್ವಾಮಿ ಆನೆ ಮಲೆಯ ಹಾಕಿಕೊಂಡವರೇ ಮಾದೇವ, ಜೇನು ಮಲೆಯ ಒದ್ದುಕೊಂಡವರೇ. ಆ ಆ ಆ ಆ
ರಾಷ್ಟ್ರಕವಿ ಕುವೆಂಪು ವಿರಚಿತ ನಾಡಗೀತೆಯನ್ನು ಈಗ ತಮ್ಮ ಮುಂದೆ ಸಾದರಪಡಿಸಲಾಗುವುದು. ಸಭಾಸದರು ದಯಮಾಡಿ ನಾಡಗೀತೆಗೆ ಗೌರವ ಸೂಚಕವಾಗಿ ಎದ್ದು ನಿಲ್ಲಬೇಕಾಗಿರುತ್ತೆ.
ಜನನೀಯ ತನುಜಾತೆ ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ, ಜಯ ಸುಂದರ ನದಿವನಗಳ ನಾಡೆ, ಜಯ ಹೇ ರಸ ಋಷಿಗಳ ವೀಣೆ. ಭೂದೇವಿಯ ಮಕುಟದ ನವಮಣಿಯೇ, ಗಂಧದ ಚಂದ್ರದ ವಂದಿನ ಗಣಿಯೇ, ರಾಘವ ಮಧುಸೂದನರವತರಿಸಿದ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಜನನಿಯ ಜೋಗುಳ ವೇದದ ಘೋಷ, ಜನನಿಗೆ ಜೀವವು ನಿನ್ನಾವೇಶ. ಹಸುರಿನ ಗಿರಿಗಳ ಸಾಲೆ, ನಿನ್ನಯ ಕೊರಳಿನ ಮಾಲೆ. ಕಪಿಲ ಪತಂಜಲ ಗೌತಮ ದಿನನುತ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಚಂದ್ರಮ ವೈಸಳರಾಳಿದ ನಾಡೆ, ಕಂಕಣ ಜಗಣರ ನೆಚ್ಚಿನ ವೀಣೆ. ಕೃಷ್ಣ ಶರಾವತಿ ತುಂಗ, ಕಾವೇರಿಯವರ ರಂಗ. ಚೈತನ್ಯ ಪರಮಹಂಸ ವಿವೇಕರ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಶಂಕರ ರಮನುಜ ವಿದ್ಯಾರಣ್ಯ, ಬಸವೇಶ್ವರ ಮಧ್ವರ ದಿವ್ಯಾರಣ್ಯ. ರಂಗ ಷಡಕ್ಷರಿ ಪೊನ್ನ, ಪಂಪಲ ಭೂಮಿಪತಿ ಜನ್ನ. ಕುಮಾರ ವ್ಯಾಸರ ಮಂಗಳ ಧಾಮ, ಕವಿ ಕೋಗಿಲೆಗಳ ಪುಣ್ಯಾರಾಮ. ನಾನಕ ರಾಮಾನಂದ ಕಬೀರರ ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ. ಸರ್ವ ಜನಾಂಗದ ಶಾಂತಿಯ ತೋಟ, ರಸಿಕರ ಕಂಗಳ ಸೆಳೆಯುವ ನೋಟ. ಹಿಂದೂ ಕ್ರೈಸ್ತ ಮುಸಲ್ಮಾನ, ಪಾರಸಿಕ ಜೈನ ಉದ್ಯಾನ. ಜನಕನ ಹೋಲುವ ದೊರೆಗಳ ಧಾಮ, ಕಾಯಕ ವೈಣಿಕರ ರಾಮ. ಕನ್ನಡ ನುಡಿ ಕುಣಿದಾಡುವ ಗೇಹ, ಕನ್ನಡ ತಾಯಿಯ ಮಕ್ಕಳ ದೇಹ. ಭಾರತ ಜನನಿಯ ತನುಜಾತೆ, ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ, ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ, ಜಯ ಹೇ ಕರ್ನಾಟಕ ಮಾತೆ. ಧನ್ಯವಾದಗಳು.
ಪ್ರಾರ್ಥನೆ ಹಾಗೂ ನಾಡಗೀತೆಯನ್ನು ಹಾಡಿದ ಡಾಕ್ಟರ್ ನಿಂಗರಾಜು, ಡಾಕ್ಟರ್ ಮಧುಸೂದನ್ ಹಾಗೂ ಡಾಕ್ಟರ್ ಲೋಕೇಶ್ ಅವರಿಗೆ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ವಂದನೆಗಳನ್ನ ಸಲ್ಲಿಸ್ತೇನೆ. Today we have amongst with us Dr. K. Namrutesh, a notable professor at the Department of Studies in Botany and past chairman of the department. Dr. Namrutesh is a distinguished fellow of Karnataka State Science and Technology Academy and the National Academy of Biological Sciences. Dr. Namrutesh has earned a doctoral degree from the University of Mysore with a prestigious ICAR fellowship and a postdoctoral degree from the USA. His leadership as the IKC director underscores his role in maintaining academic excellence within the university. Additionally, Dr. Namrutesh serves as the coordinator of the University Endowment Lecture Organizing Scheme, which brings us together for this significant event today. It is now my honor to invite Dr. Namrutesh to deliver the welcome address.
ಎಲ್ಲರಿಗೂ ನನ್ನ ನಮಸ್ಕಾರಗಳು. ಎ ವೆರಿ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಎವ್ರಿ ಒನ್. ಆನರಬಲ್ ವೈಸ್ ಚಾನ್ಸಲರ್ ಪ್ರೊಫೆಸರ್ ಎನ್ ಕೆ ಲೋಕನಾಥ್, ಟುಡೇಸ್ ಚೀಫ್ ಗೆಸ್ಟ್ ಡಾಕ್ಟರ್ ಅನಿಲ್ ಕುಮಾರ್ ತ್ರಿಪಾಠಿ, ಡೈರೆಕ್ಟರ್ and senior professor Banaras Hindu University, another distinguished alumnus of the University of Mysore, Dr. S.R. Niranjana, Vice Chairman, Higher Education Council, and Administrative Head, Mrs. V.R. Shailaja, and respected Chairman of the Department of Botany, Dr. Rajkumar Garampalli, special invitees, students community of the Life Sciences, and also from Bharati College, Bharati Nagara, Maranis, and JSS College, and other special invitees of the today's function, media personnel. It gives me an immense pleasure to again signify an important event after a gap of 10 days. That is the University Endowment Lecture Program, IQAC and Department of Studies in Botany jointly organizing to commemorate or to signify the important Danish scientist Professor Neergaard and Professor Viggo Smedegaard. There is a saying in English, success is far more difficult to maintain than it is to achieve. The success was achieved by Professor K.M. Safiullah, then Chairman of the Department of Studies in Botany, the maintenance is very difficult. That has been carried by three important dignitaries on the campus: Professor S. Shekhar Shetty, Professor H. S. Prakash, and another important dignitary who is on the dais, Professor S. R. Niranjana. They have brought to the Department of Applied Botany and Biotechnology to the international stature. Because of the visit of Professor Paul Neergaard under Sir M. Visheshwaraya chair, that's really has turned out a big point in collaborating between the University of Mysore and Danish Government Institute of Seed Pathology and the Royal Veterinary Agriculture University, Copenhagen, Denmark. This has led to the establishment of Pulmonary Downey Research Laboratory, and subsequently the great contribution of Professor S. Shekhar Shetty and especially two important personalities, Dr. S. R. Niranjana and Dr. S. S. Prakash, for the establishment of Asian Seed Pathology Center at the Department of Applied Botany and Biotechnology. So, to mark the contributions of these two important Danish scientists, our professor mentor Dr. S. Shekhar Shetty instituted the endowment on these two important personalities. So that is why we are witnessing again after Professor S. Shekhar Shetty endowment lecture today. So with this backdrop, I am extremely happy to welcome our Honorable Vice Chancellor, who is always inspiring the campus with academic excellence and bringing financial discipline on the campus after assuming the charge of Vice Chancellorship. As the director of PMEB, he was responsible in bringing the NIRF rank for the consecutive 4 times below 50. That is the greatness of our Honorable Vice Chancellor, Professor N.K. Loknath. So with this backdrop, I sincerely requested Dr. J.R. Janardana to welcome our Honorable Vice Chancellor to this specially arranged endowment lecture of two Danish scientists. Sir, I welcome you, sir. Another important dignitary is on the dais, Professor S. R. Niranjana, Vice Chairman and former Vice Chancellor, Gulbarga University. ಅನ್ವಯಿಕ ಸಸ್ಯಶಾಸ್ತ್ರ ಮತ್ತು ಜೈವಿಕ ತಂತ್ರಜ್ಞಾನವನ್ನು ಬಾನೆತ್ತರಕ್ಕೆ ಕರ್ಕೊಂಡೋದಂಥ ಕೀರ್ತಿ ಅವರಿಗೆ ಸಲ್ತದೆ ಅಂತಂದರೆ ತಪ್ಪಾಗಲಾರದು. ಡಾಕ್ಟರ್ ಶೇಖರ್ ಶೆಟ್ಟಿ ಅವರ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಬೆಳೆದಂಥ ಇವರು ಅತ್ಯಂತ ಹೆಚ್ಚಿನ ಮಟ್ಟದಲ್ಲಿ ಅಂದರೆ ಸುಮಾರು ಇವತ್ತು ನಡೀತಿರ್ತಕ್ಕಂಥ ಡ್ಯಾನಿಡಾ ಪ್ರೋಗ್ರಾಮ್ನ ಎಲ್ಲ ಮಜಲುಗಳಿಗೆ ಸುಮಾರು 40 ಕಂಟ್ರಿ ಒಟ್ಟಿಗೆ ಕೊಲಾಬ್ರೇಷನ್ ಮಾಡ್ತಕ್ಕಂಥ ಅವಕಾಶನ ಅವರು ಈ 3 ಜನ ಮಾಡಿದ್ದಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ ತಪ್ಪಾಗಲಾರದು. ಅಂಥ ವ್ಯಕ್ತಿತ್ವದ ಡಾಕ್ಟರ್ ಎಸ್ ಆರ್ ನಿರಂಜನ್ ಅವರು ಇವತ್ತು ಉನ್ನತ ಶಿಕ್ಷಣ ಪರಿಷತ್ನ ಉಪಾಧ್ಯಕ್ಷರಾಗಿಯೂ ಸಹ ಅತ್ಯಂತ ಯಶಸ್ವಿಯಾಗಿ ಗುಲ್ಬರ್ಗ ವಿಶ್ವವಿದ್ಯಾಲಯದ ಕುಲಪತಿಗಳಾಗಿ ಸೇವೆ ಸಲ್ಲಿಸಿ ಸಂಶೋಧನಾ ಕ್ಷೇತ್ರದಲ್ಲಿ ತಮ್ಮದೇ ಆದಂಥ ಕೊಡುಗೆಯನ್ನು ನೀಡಿರ್ತಕ್ಕಂಥವರು ಇಲ್ಲಿದ್ದಾರೆ. Dr. Anil Kumar Tripathi avrna is responsible in bringing Dr. Anil Kumar Tripathi FNA, an eminent scholar of the nation and international repute. Dr. Anil Kumar Tripathi, who is our Dr. S. R. Niranjana is responsible, who is here. I sincerely request Dr. N.S. Raju to welcome Dr. S. R. Niranjana on this occasion, because who is responsible in holding this important endowment lecture. Sir, welcome you, sir, to this function, sir. Please. So when I received the direction from our Honorable Vice Chancellor, Professor N.K. Loknath, to invite Dr. Anil Kumar Tripathi, so who is an eminent scholar, especially he has worked on signaling molecule, especially the Rho factor, so especially the plant growth promoting rhizobacteria, which is considered, and plant growth promoting rhizosphere fungi, which are considered as the defense personnel of any country. The microbiota, especially beneath the rhizosphere soil, which are important, they are playing pivotal role. He has conducted all the research activities under that PGPR by taking only one organism, Azospirillum brasilense, that he is going to deliver an important aspect. How the carbon source utilization is helpful in the Rhizocompetence. That's instead of going for green revolution, with due respect to Dr. Narmani Borlag and Dr. M.S. Swaminathan, we wanted evergreen revolution, for which Dr. Tripathi is responsible to take the nation to the forward by going for evergreen revolution. In green revolution, NPK, the nitrogen, NPK chemical fertilizer has been poured into the soil, which actually diminished the quality of this soil. But here is the person who is taking care of plant growth promoting rhizobacter, an important characteristic feature of the beneath the soil, that is the microbiota or microbiome, who is responsible in conducting various activities. You can take out the details of biodata will be known to you by our colleague Dr. J. Lohit while felicitating him on behalf of University of Mysore. Sir, we are fortunate enough to have you, sir, on this morning occasion, sir. Really, we are proud because he is from BHU. Both University of Mysore and BHU, Banaras Hindu University, started together. And Chancellor for both the universities, our Nalvadi, His Excellency Nalvadi Krishnaraj Vadera. That is the greatness. Both the universities started, University of Mysore, and both the universities are 108 years old, and we are like brothers. So such a wonderful atmosphere that we are having, sir, because of your presence. Really, we are honored to have you on this occasion, sir. On behalf of University Endowment Lecture, University of Mysore IKAC, and I request Dr. MP Sadashiva to welcome Dr. Anil Kumar Tripathi on this occasion. Please, sir, please, a big round of applause for accepting. He has come all over from Punjab, Mohali to Mysore University, sir. Welcome, sir. Another distinguished guest of the day, administrative head, an alumna of Department of Studies in Geography, who is meritorious all through her career. She is here, and very inspiring to all the girl student. She is here. She is a senior most KS officer who is here, who is always backbone of any program for that matter. So I sincerely welcome. I request our office staff to welcome the Mrs. V.R. Shailaja on this occasion, sir. Welcome, madam. So when we received the directions from Honorable Vice Chancellor to conduct the second endowment lecture, so after a gap of 10 days, actually we conducted 24th Dr. S. endowment lecture, instituted by private company, Pioneer Seed Company. Again, see the visionary aspect of Dr. S. Shetty in association of Department of Studies in Botany as a value. Because Dr. K.M. Sapiola, he had invited Dr. Paul Neergaard and Sir M. Vishweshwara. He knew the importance of the endowment, chair and everything. That's why he instituted by spending 5 lakhs in the name of Professor Paul Neergaard and Professor Viggo Smedegaard. Until he has visited to the Department of Studies in Botany at that time when Dr. K.M. Sapiola was the chairman. The respected chairman, Dr. Rajkumar H. Garampalli, when I contacted him, all senior faculty, Dr. J.R. Janardana is here, who is also the recipient of Paul Neergaard Gold Medal, who is also here, unanimously accepted our association to have the association with us, for which he is responsible here. He is also very actively participating by involving in arranging all these activities. Sir, I welcome you, sir. Sir, for this function, I request Dr. J. Lohit to welcome him on this occasion, sir. Please, sir. So, I also welcome former Vice Chancellor Professor P. Venkatramaya and the institution builder of Coimbatore University, who recruited. Even today they are working functionally very well. So who is also very supportive, and he has established a very good program in the University of Mysore. Even today he is working as the chairman of the school-going children to introduce the science program. He is so active. We have to be following him for our inculcate the activities whatever he is doing today. So eminent physicist who is also here witnessing, and another person, my mentor, Dr. S. Shetty, an institution builder who is here, who is responsible to hold this important event on the campus. It will take another 50 years because of this endowment, because Dr. Anil Kumar Tripathi is here. We might have another collaboration. That is the vision of Dr. S. Shetty. I also welcome you, sir. Dr. H.S. Prakash and Dr. G. Subramania, Dr. J.R. Janardana, Dr. M.P. Sadashiva, Dr. N.S. Raju, Dr. Sita Naik, Principal of Yuvarajas College, Dr. M.K. Mahesh, Dr. M.N. Anil Kumar, Dr. B. Sandappa, and students community from on the campus, from Department of Studies in Botany, then the zoology, microbiology, environmental science, the genetics and genomics, molecular biology, and students very actively participating from Bharati College, Bhatnagar, my alma mater, and JSS College, Ooty Road, and the Mahajanas College, Monarch College, everyone is, all the botany fraternity are here to witness an important event on this occasion, sir. We are honored on behalf of University of Mysore. We sincerely welcome you all for this important participating in this important event. I also welcome ICT people and also media personnel, especially for covering the program very well for the Mysore city. ಸೊ ಒನ್ಸ್ ಅಗೇನ್ ತಮ್ಮೆಲ್ಲರಿಗೂ ವಂದಿಸುತ್ತಾ ಒಂದು ಒಳ್ಳೆ ವಿಶೇಷ ಉಪನ್ಯಾಸಕ್ಕಾಗಿ ಕಾಯೋಣ. ಒಳ್ಳೆಯದಾಗಲಿ ಅಂತೇಳಿ ತಮಗೆಲ್ಲರಿಗೂ ಶುಭ ಹಾರೈಸುತ್ತಾ ನನ್ನ ಕೆಲಸವನ್ನು ಮುಗಿಸ್ತೇನೆ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
ಎಲ್ಲರನ್ನ ಆತ್ಮೀಯವಾಗಿ ಸ್ವಾಗತಿಸಿದಂತಹ ಪ್ರೊಫೆಸರ್ ಅಮೃತೇಶ್ ಅವರಿಗೂ ಕೂಡ ತಮ್ಮೆಲ್ಲರ ಪರವಾಗಿ ಹೃತ್ಪೂರ್ವಕವಾದ ಸ್ವಾಗತವನ್ನು ಕೊಡ್ತೀನಿ. Dr. Amritesh in his welcome address has highlighted that this endowment lecture was instituted under the visionary leadership of Professor Shekhar Shetty. Now, it is with great honor that I invite Professor S.R. Niranjana, the Vice Chairman of Karnataka State Higher Education Council, to inaugurate Professor Neergaard and Professor Viggo Smedegaard Endowment Lecture Program by lighting the ceremonial lamp. I also invite other dignitaries present on the dais to join hands with Professor Niranjana in this act. ಕಾರ್ಯಕ್ರಮವನ್ನು ವಿದ್ಯುಕ್ತವಾಗಿ ಉದ್ಘಾಟಿಸಿದ ಗಣ್ಯರಿಗೂ ಕೂಡ ವಂದನೆಗಳನ್ನ ಸಲ್ಲಿಸ್ತೀನಿ. Ladies and gentlemen, it is my distinct honor to introduce Professor S.R. Niranjana to this august gathering. Okay, sir. With an illustrious career spanning over 35 years, Professor Niranjana has mentored 21 doctoral students and authored over 213 research articles, boasting an impressive H index of 42 and over 5,600 citations. It is now my honor to invite Professor S.R. Niranjana to deliver the inaugural address, setting the stage for an enlightening session.
So good morning all of you. Ellargu namaskara. The University of Mysore and the Internal Quality Assurance Cell is organizing a endowment lecture on the Professor Neergaard and Professor Viggo Smedegaard Endowment Lecture Program. On this occasion, the Honorable Vice Chancellor of Mysore University and good friend of mine, Professor N.K. Loknath, and another invited a distinguished guest. He is agreed to deliver the lecture on the occasion of Neergaard and Viggo Smedegaard Endowment Lecture. Professor Anil Kumar Tripathi is a director of the Indian Institute of Science and Education and Research ISR, Ministry of Education, Government of India, Maholi, and is also a senior professor of Department of a School of Biotechnology in Banaras Hindu University, Varanasi. And another guest on the dais, very respected administrator and young, enthusiastic Shailaja Madam, she is on the dais, and then the head of department of botany, Dr. Rajkumar, and the very enthusiastic coordinator of this endowment program, Professor K. Amrutesh, and a senior most, a former Vice Chancellor, Professor Venkataramaya, and Professor Shekhar Shetty, Professor Prakash, Professor Subramania, Professor Janardhan, Professor Raju, and Professor Sadasiv, and other invited guests, and the students, media personnel, ladies and gentlemen. We have a very short time because I curtailed my biodata to read the people who are on the dais. And Vice Chancellor is ready to go to some other program. And I have to rush to the 4 o'clock Chief Minister meeting. And we have a less time. So this is all the very recent developments. And the Chief Guest, Dr. Anil Kumar, has to go for a meeting by 4 o'clock in Bangalore. So instead of taking too much of formalities, the University of Mysore, in the short span of time, Dr. Amrutesh has organized a 2-lecture endowments. So last week we have organized Professor Shekhar Shetty endowment lecture in the same hall, and now immediately after 1 week or 10 days, we are organized this Paul Neergaard and the Smedegaards. My dear students, it is a very rare occasion of the universities to have a foreign scientist endowment in the university system. So Professor Shekhar Shetty has taken keen interest to introduce the endowment in the university long back, and these lecture endowment is not held because of many reasons in the university. But now, as a faculty of the university, I decided that it has to be organized. Last time also I told the Honorable Vice Chancellor, endowment has to be run very regularly and inviting the man like Professor Anil Tripathi. The endowment lecture should be delivered by a very distinguished scientist of this country. So that should be the justification for the endowment. I am not telling that everybody can deliver lecture. I can also deliver, anybody can deliver lecture. But endowments having their own kind of a prestige and privilege of the people who are contributed. As Amrutesh mentioned that who is these two people? These two people are supported our department for long term. Paul Neergaard was the visiting professor in Vishwesvaraya chair in Mysore University. I think now nobody is there in the Vishwesvaraya chair. I think Vice Chancellor has to keep interest and register also you keep somebody in that chair to work for the university upliftment. That is invited after 10 years, I know that. But the thing is that regularly it has to be done. Regularly somebody has to work. And long back one man was there, Gopinath from VRC. After that nobody, I know. So that this Paul Neergaard, you know that he is a well-known sheep pathologist of the Royal Veterinary and Agriculture University, Copenhagen. Earlier it was called as a DGSP. All we are trained and visited that institute. And nearly about 70 to 80 scientists are invited from this country to train in the area of sheep pathology. Neergaard was a very famous sheep pathologist, and you know that the first book was Sheep Pathology by Paul Neergaard, was very famous book. Nobody has written so far the interception of seed-borne diseases from one region to another region. The last chapter of this book is indicating that how the seed-borne pathogens, how the seed-borne diseases are intercepting from one country to another country. That's what this interception has taken place by the quarantine. So Paul Neergaard was given very nice net cell in that book in the last chapter. It helped many people in the quarantine for the exchange of pathogens and virus bacteria. That book is written by Paul Neergaard. And later on, after that, Professor Shetty has developed a bridge between the Copenhagen Veterinary University and the Department of Biotechnology and Applied Botany in University of Mysore for the Viggo Smedegaard is another scientist. He came later to work with us as a collaborator on the plant disease resistance. So that these two people are contributed for 30-40 years for our department and many of the students are trained in this program. They are benefited out of this program. That's what the Professor Shetty is our good a professor respected in the department. He has taken us in a very nice manner in academic way, and he has instituted this endowment lecture. That is the importance of the endowment. Today, the university is organizing this endowment lecture. I am very happy that at least we are now remembered after many years to organize this endowment. My dear students, as chairman, as vice chairman of Karnataka State Higher Education Council, I will take a few minutes to highlight. I will not speak science today. I will speak what is the policies are there in the Karnataka government has initiated so far in the education system. How educations are uplifting in the university. What is the role of education council? Very few states are having this kind of council. So higher education council recently, very few states have started. In that, Karnataka is the one very active in the making the policies and advising the universities. Karnataka State Higher Education Council is an advisory body between the government and the higher education institutes. All your NAPR, SCP, your fee structure, your academic calendar, your syllabus, your credits, everything is going to be decided in the council. So that and quality, Karnataka State Higher Education Council was set up to synergic relationship among the government and universities, academics, experts, by occupying the operational space between the government and the universities on one hand, and the other hand between the university and apex level regulatory bodies on the other hand. So this is the main duty of the regulation. So that conducting examinations, recently we have received a very serious letter that I think I will be circulated to all the vice chancellors, and result has to be announced well in time in the universities. Students at any cost should not deny by any opportunities of the further future aspects. That's what I requested all the vice chancellors to announce the results in well in time, so that we can make the students to get the good kind of opportunities for their future. The Karnataka is having a 360 government degree colleges. There are 32 public universities, and including a medical and agriculture, we have a 22 lakh students in Karnataka. It is a hub. Karnataka became a hub for the higher education today. High number of universities, 27 private universities, 32 public universities, 443 the government colleges. All together we have a 900 so far the colleges, and we are giving a more scope on higher education, making the lot of efforts, providing the quality higher education, ensuring accountability, and then making prospective plans for governance and higher education with pertain to the administration, affiliation, recognition, and inspections. Accountability of the students' learning, accountability of the conducting examinations, accountability of the administration, accountability of the affiliation. All these things are very important. Government has given more importance on that one. And after assuming charge, I have taken all these things. Affiliation is well in time, and recognition of the private universities, inspections of private universities and public universities is going very well. And we are insisted all the universities and colleges who are not subjected to the university for accreditation for NAC, and they have to do it immediately for their accreditation for the NAC. And recently we have signed 11 MOUs with foreign universities. Recently, last 1 year, the government has taken initiative and signed up 11 universities. And I am repeatedly telling that all the universities have to take part in that. And recently I visited in UK universities. I told Professor Lokanath, physics is the one area they are very happy to collaborate. All the 3 universities we have selected, Dharwad and Mysore and Bangalore, for the physics collaboration, especially in the area of electronic and electrical, and the physics and astronomy. Then semiconductor, clusters, these areas. Life science, I am going around now, life science are somehow somewhere it is declining the collaboration because of the lack of scientists come forward to take the collaboration. That you have to very seriously think that one. And this MOIC is going to help in future for many universities. And curriculum structuring, recently we have given the curriculum structure for the NAP and SCP. You don't confuse anything among this SCP or NAP, nothing is there. We are revised the course, but you are crossed now. Anybody sitting in the undergraduate, you need not worry about that. we are retaining the credit as per the UGC guidelines and we restructure the curriculum on par with the UGC guidelines. Don't be under that any ambiguity or any apprehension for the NEP or SCP. We are not ready to trouble any students. We are taking it in a very smooth way. That we have done that in the last recent time, and we have told that, and how many credits and everything else we have given in a very right manner. It is taking place. And recently, the British Council has come forward to conduct the English teaching training program. And this British Council is giving an English training program for the teachers to learn English. That we are not thinking that our teachers are inferior in teaching in English. But communication English, our students are not able to compete in the communication of the competitive examinations. That was I am telling that all the students take that confidentiality and your courage in facing the competitive examination. You are not. The Indian students are not competing properly in competitive examination. I don't know the reason behind that. You assess yourself why you are not able to compete with the competitive examinations. If competitive examinations are not, you are regularly facing this one. If in the future course of time, and I have sent one circular, this year every university has to conduct the entrance test for the PG admissions. Every university. Now the Bangalore is not conducting, Mysore is conducting, Gulbarga is not conducting, Dharwad is conducting. So these things are there, one university is conducting, one university is not conducting. Now we made it very compulsory. This academic year, every university has to conduct the entrance examination. And recently, you know that we have proud 6 universities has got the PM Usha grant from the central government on the S60-40 ratio. 40% is the government and 60% is the state government is the 40%, 60% is central government. And that 2 universities they have got the 100 crore, Bangalore University and Rani Chennamma University. Another 4 grants, because don't worry that this university has got 100 crore long back. Our academic credentials already we have taken, but now that I am insisting the Vice Chancellor to go for some other programs where we can get the grant and sustain the financial discipline and financial upliftment in the university, and we have to think more. And the 4 universities have got the 20 crore grant. One is the Marani Cluster University, another is Gulbarga, another is the Dharwad, and another is the Mangalore. And universities are competing now. This is the time to compete for the upliftment of the quality education. And for upliftment of quality education, we should not depend on the government always. Government is not always supporting the university. University and students have to struggle to create a grant themselves where the fund is available. We have to make our academic credentials. We have to contribute and make record and document it and submit for NRF ranking, submit for NAC upliftment. These are all the assessments is going to help the universities. So totally 5 colleges and 3 districts, we got this time 413.96 crores on the 60-40 ratio from the central government. So that we are proud in making that kind of the amount we got it to the state. Now the state government is thinking that to support the universities, and now we have started the skill orientation program is the important area that the government is concentrating. So recently, Mysore we were not selected because of many reasons. We are selected Davangere, we are selected Tumkur, we are selected Raichur, and we are selected Gulbarga. 4 universities we have started the pilot experiments of introducing a skill-oriented course for BA, BSc, BCom programs, and these BA, BSc programs, the students are offered skill program, industries are come forward, and Karnataka Skill Development Corporation is paying that amount. Universities need not to pay for that courses, and we are going to give the skill orientation program. In the next year onwards, all the universities we are considering and making the budgetary allocation. Chief Minister has told that we will make the budgetary allocation for all the universities, and all the undergraduate students are getting the skill oriented courses in your education system. So the government is taking all these things. Now we have to work on this one, and a tough job for the universities. Competition is there. We have to struggle to uplift the universities, students to excel well, especially in all the areas. Not only your regular result, you have to excel in your research, you have to excel in your publication, you have to excel in your areas of research, what we are going to do. All these areas is very important, and now this is the stage university has to take the many initiative. And recently I got the paper that I think I am going to circulate this paper. You should know that they have got the percent trusted to tell the truth. So that has been published in a graph, and scientists, we are in the place of 1, 2, 3, 4, 5, 6. Teachers are in the 6th position, scientists are in the 9th position, nurses are 1st position, and aeroplane pilots are the 2nd position, librarians are 3rd position. So how we are trust the profession? Scientists we are in the 9th position, teachers are in the 6th position, and the politicians in the last position. So it has been recorded here. The percentage was given 9% for the politician, 88% for the nurses. See, nurses, they are the commitment. That's what they are rated 99%. And the scientists, librarians are rated 86%. Teachers are rated 70, teachers are rated the 78%. Professors are rated 76%. Scientists are rated 74%. Rest of the things, they are civil servants and everything as mentioned. And this data is telling that the commitment of a kind of work you are doing. Fortunately, unfortunately, the students are not rated here. In future, students are going to rate how committed they are for their education. How committed they are to do their education. That's very important for that. Year afterwards, it is going to be assessed like this. Then we should know where our position, what is our duties to come up with the higher percentage. So everything has been statistically analyzed. Everything has been statistically analyzed. Government is working on that one. Government is very keen in uplifting the higher education in the state, making too many attempts, and the skill courses are a priority of the government. With these words, I thank once again Professor Tripathi accepting my invitation. I was given a invitation 1 month back. He is very busy schedule. He is working in many the bodies, and he is in the, as per this ranking, people also assess the Tiwari post as director of this institute, where the science and education in Mawoli, he is in the 17th position as director as far as administration is concerned. Like that, the vice chancellors, directors, registrars, everybody was rated in the level of your assessment. So year after the country is going to assess in that way. Nobody is superior, nobody is inferior. So everybody is accountable for this kind of assessment. This assessment will help for us to improve ourselves. So scientist has to work on that one to come to the place of a nurses, and the professor has to work more to come up with the next the graphing sale. And these are all the things are going to do that. And I thank once again for the Tripathi especially accepting all the way he travelled from Chandigarh to Mysore. And see, his experience is going to be shared with you as a teacher, and he is serving as an administrator, and he is sitting as an administrator in central government, and we are expecting more from his contribution. So the administrator has to work more. Loknath has to not to do science now. Not to do science now. He has done enough science now. Now he has to make the people to work as per science. That is important for a leader. He is an academic leader. Any vice chancellor do not. He is taking a class. I know he is going to department every day. He is taking class and interacting with teachers. I appreciate it. When the council was organized the lecture series of every month, how many students you are logging in that lecture? I don't know. You do it. And PMEB board, I insist in the registrar to recall him and tell that link has to be circulated to all the students. Every month one university is giving a lecture on the different disciplines, all professors. I made it that regular in every month the last week. This month I think Kannada University, we are going as per the seniority of the university. Recently Shimoga we are completed. Now this month on 30th, a Kannada University is delivering, but we will consider Kannada also. Now the Kannada is very important for everybody, and that Kannada this is there, next in this university. Take that login and listen the lectures. What is going in that particular university? So I made that attempt to as a research day. Research lecture day, we called it in the government, and we are coined that word. So you take up the advantage of all the lectures, and in that lecture, first university was Mysore, and I am very happy. Government is also happy. The Vice Chancellor himself has delivered a lecture. First lecture was Loknath was delivered in that lecture series day. So I appreciate his interest, and I am requesting more and great people to work in the university. And I thank Professor Venkatramaya and Professor Shetty has made the time for this endowment lecture series and sitting here and seeing the things are going in this second. Once again, I thank all of them for their patience here. Thank you very much.
Thank you, sir. There is a slight change in the schedule. On behalf of the organizers, I now request our Honorable Vice Chancellor to felicitate Professor Anil Kumar Tripathi, the Director of IASCR. dear esteemed members of the audience, we have amongst us Professor N.K. Lokanath, is a distinguished physicist and a material scientist who has made remarkable contributions to his field through pioneering research, dedicated teaching, and exemplary leadership. Today it is a distinct privilege to listen to his presidential address. Without delay, I respectfully invite our Honorable Vice Chancellor to address this learner gathering.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ವೆರಿ ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಟು ಒನ್ ಆ್ಯಂಡ್ ಆಲ್ ಹಿಯರ್. Dignitaries on the dais, Dr. Anil Kumar Tripathi, the chief guest of the today's function, director of ISER, Mohali, Professor S.R. Niranjana, Vice Chairman, Karnataka State Higher Education Council, and my colleague Shailaja, the registrar, and the director IQSA, Professor Amrutesh, and Professor Rajkumar Garampur, the chairman, Department of Studies in Botany, and my guru, Professor Venkatramaya, and Professor Shekhar Shetty, Professor Prakash, Professor Subramania, and all senior professors, all my faculties, research students. It's a great occasion for us to heard about this very well-known endowment lecture instituted by the Dr. Shekhar Shetty, and I know that Professor Niranjana sir as well as the Professor Amrutesh has explained about how this endowment lecture has initiated in our universities. We should have to thank Professor Shetty as Department of Applied Botany and as well as now the Department of Biotechnology has made this endowment lecture is possible. So as students are here, there are two things are there. The important thing of endowment lecture is, the first thing is we need to know what is going on the present technology or in your research field on your subject. The second thing is we need to have an interaction with people and we need to collaborate with people so that we can able to establish better relations and better understanding of science. For that, this type of endowment lecture has been initiated. I urge all the students to utilize this and try to think that how it is possible. And Professor Tripathi is the one who is a well-known person, and all his busy schedule, he came all the way from Mohali to here to enlighten you. Hopefully, you will learn this lecture, and I hope all the faculties also get benefited. And also, as Professor Niranjana sir mentioned that about the university and other things, and also I request the government has to take initiation steps to recruit the faculties. Without faculties, how university will grow? In the same way, so the financial problems, most of the universities, particularly state universities, are having some problems. I urge the Karnataka Higher Education Council at least try to solve these issues as early as possible. I do not take much time because we are both having a meeting at 11 o'clock as a vice chairman, so I need to attend the same meetings. So I wish all the students should learn from good things from Professor Tripathi so that he will learn and he will also teach how it is important, the plant science. So in this, with this I am also very expecting that most of the research students look into his research work and try to follow him. And the other thing is, whenever you have a lecture, I have seen most of the students are in between. You take one notebook and make a notes. After 10 years, if you are in the research field or in a teaching field, you will remember how Tripathi has been prepared the lecture and how he prepared. At least you should know that. So with that, thank you for giving me an opportunity to come over here, and all the best. Good luck. Thank you, Nanda.
ಧನ್ಯವಾದ ಸರ್. ಈಗ ಆಯೋಜಕರ ಪರವಾಗಿ ಇಂದಿನ ಮುಖ್ಯ ಅತಿಥಿಗಳು ಹಾಗೂ ಸಂಪನ್ಮೂಲ ಅತಿಥಿಗಳಾದಂತಹ ಪ್ರೊಫೆಸರ್ ಅನಿಲ್ ಕುಮಾರ್ ತ್ರಿಪಾಠಿ ಅವರಿಗೆ ಗೌರವ ಸಮರ್ಪಣೆಯನ್ನು ಸಲ್ಲಿಸಬೇಕಾಗಿ ಸನ್ಮಾನ್ಯ ಕುಲಪತಿಗಳನ್ನ ವಿನಂತಿಸ್ತೇನೆ. Ladies and gentlemen, it's with great pleasure that I stand before you to introduce a distinguished academician and researcher, Professor Anil Kumar Tripathi. With over 4 decades of teaching experience, Professor Tripathi has been a stalwart at Banaras Hindu University, where he has not only imparted knowledge but also nurtured countless young minds in the field of biotechnology. His leadership roles have been pivotal, serving as director of the Institute of Science at BHU and steering the ship at the Central Institute of Medicinal and Aromatic Plants. Currently, Professor Tripathi stands at the helm of the Indian Institute of Science Education and Research, a role he assumed recently. His leadership continues to pave the way for cutting-edge research and transformative educational initiatives in the realm of biotechnology. Professor Tripathi's research interests are as diverse as they are impactful, focusing on bacterial genetics, functional genomics, systems genomics, and synthetic biology. His pioneering work has not only advanced scientific knowledge, but also laid the groundwork for groundbreaking discoveries in these critical areas. In recognition of his outstanding contributions to science and academia, Professor Tripathi has been bestowed with numerous prestigious awards and fellowships. These include the J.C. Ghosh National Fellowship, the fellowships from the esteemed bodies such as the Indian Academy of Sciences, Indian National Academy, Indian Academy of Sciences India, and many more. Professor Anil Kumar Tripathi epitomizes dedication, scholarship, and leadership in the field of biotechnology. His commitment to scientific inquiry and academic excellence serves as a beacon of inspiration for us all. Therefore, it is with great honor and respect that I now invite Professor Tripathi to deliver today's endowment lecture.
Esteemed Vice Chancellor in absentia, Professor Niranjana, the Vice Chairman of Karnataka State Higher Education Council in absentia, the Registrar, Head of the Department, and very eminent teachers sitting in the front rows, the architects of this department and its academic program, the faculty members, students, and friends. I feel extremely delighted and honored to be here in this institution and this city, which is rich of traditions, history, and legacy. Because, as was said, that Banaras Hindu University and the University of Mysore were founded at the same time, 1916, the same year they were founded. And I think the vision of both the institutions, the founders of both the institutions, Pandit Madan Mohan Malviya and Sri Vishweshwaraya, this vision were also ahead of their time. Because when the leaders of the country were struggling for the independence, these academic leaders were thinking that what after the freedom? Once the freedom is attained, what will be the future of the country? When the British were telling that Indians are not capable of ruling themselves, they need British rule. At that time, these two gentlemen were working to prove that the Indians are not only capable, they might eventually be better than the Britishers. And with that kind of spirit, these institutions were established to generate human resource, to produce manpower, which will help in the rebuilding of the nation and creating a kind of global leadership which the present leadership is aspiring for. When I was invited by Professor Neergaard to deliver my talk, I chose a topic, but when I see here the audience, I find very bright young faces with a lot of optimism in their eyes. And I feel that I should have probably chosen a slightly different topic. But anyway, having been a teacher for about 4 decades, I will try to simplify what message I want to give from my presentation. But before that, I would like to share a few things about my journey, academic journey from my student days. Some days back, somebody was asking me about the gender diversity in STEM, science, technology, engineering, and management. And when I see the audience today, I see huge difference in the diversity. Because all over the country we are talking about that how to increase the number of women in education. But when I look at the faces in the hall, I feel that the population of female is much higher than the males present in this house. I have to search the male faces. That's very heartening. And anyone in my place standing here will have great hopes and great optimism for the country. That the women, the girls, they are taking lead in science and technology. Why science is important? I will digress a bit. Why studying science is important? After our class 12th, our parents tell that go to engineering, go to medicine. These were the 2 major fields people were going. And no parents were encouraging their wards to choose science as their career. Because I think few decades back, this was the fundamental question, that earning bread and butter for yourself. The pursuit of knowledge, pursuit of inquiry, pursuit of self-satisfaction was not the goal. Although, more than 2 decades ago, we had great stalwarts who had great passion for the pursuit of knowledge, pursuit of science, and that is why we stand today at this level of the science and technology. You know the technology, what is the base of the technology? The base of the high-end technology is the high-end science, because all the technologies emerge from science. And if you want to have high-end technology developed, if we want our nation to be economically strong, we need to have capability for developing high-end technology. And that high-end technology will be developed if we excel in high-end science. And therefore, it's extremely important for us to address this need for high-end science. And all of you are here, most of you are pursuing the life sciences. And when I did my master's in 1980, I did master's in botany. At that time, biotechnology was not born in the country. Biotechnology was born in the country in 1985-86 by the creation of National Biotechnology Board of India, and afterwards it was converted into Department of Biotechnology. And in the country, 5 departments of biotechnology were established. MSc biotechnology training programs were established by Department of Biotechnology, and DSU was one of them. And Banaras Hindu University, JNU, MS University, Madurai Kamaraj University, and Pune University. These were the 5 universities where the MSc biotechnology program supported by DBT started. And afterwards, all over the country, many programs, many universities started offering the biotechnology, keeping the importance of biotechnology, keeping the scope of biotechnology in view, that these programs started. But soon after about 10 years or so, the kind of dream the students had when they pursued biotechnology degree programs, their dreams started shattering because there were not enough opportunities. And this was because of several reasons. One reason was the ban on the genetically manipulated organisms, ban on the use of genetically manipulated organisms. But gradually, all over the world, a need is being felt that genetically manipulated organisms have their future, keeping in view the increase in the global population, increase in the global demand. We need to have technologies which are faster. We cannot leave it to the nature to develop new varieties. You need to develop new varieties faster enough to take care of the needs of the society. So, the biotechnology, although graduates were there, but they were not getting jobs directly into the industry because biotechnology, there was less acceptability of biotechnology. But during COVID times, we have seen that there was a great application of biotechnology all over the world. Some of the techniques of biotechnology, they came to great aid in the diagnostics, particularly these real-time PCR, etc. Those who are familiar, they were in great demand. So biotechnology became the very important. Thereafter, I think the vaccine development, the diagnostics, etc., have taken the attention of the public. And now, all over the country, all over the world, people realize that yes, biotechnology is important for the society. Well, I will now shift gears and go back to my talk. I can go. Yeah. So when I was
He's good.
So I initially did after my master's, I did my PhD on cyanobacterial genetics, blue-green algae, nitrogen fixing blue-green algae. And after that, I went for my postdoctoral studies to Germany, where I started working on the genetic engineering of Azospirillum brasilense. And I deliberately chose Azospirillum because this is a bacterium which is capable of promoting plant growth. Many of you know that there are legumes, and legumes have the capability of fixing nitrogen in their nodules. So these legumes don't need any nitrogen fertilization, and therefore in many of the fields, the farmers are advised that there should be crop rotation. If you want to fertilize the field, there should be crop rotation. You have legume crop followed by cereals or rice or whatever, so that field gets enough nitrogen because of the legume. But the question was being asked that what with non-legumes? Because non-legumes do have bacteria associated with them, but which are the bacteria which can contribute to their nitrogen needs? And when this was being investigated, Johanna Dobriner, a German lady who was working in Brazil, she came up with the observation that in many of these C4 plants, C4 grasses in the rhizosphere, there is a bacterium called, earlier it was called spirillum, but later on it was called Azospirillum because this was able to fix nitrogen. That is why it was called azo. Azo means nitrogen. So this was called Azospirillum, and there were two species of Azospirillum lipophyrum and Azospirillum brasilense. They were described by Johann and Dobriner, and it was a great news because it gave a promise that for non-legume crops, which are many, you don't have to probably put fertilizer. These bacteria will take care of their nitrogen needs. But that was not to be because there was a huge demand of crop productivity, and these biological nitrogen fixation cannot take care of that need, and therefore nitrogen fertilizers were utilized. You might have heard about the Haber-Bosch process. How many of you know among the students what is Haber-Bosch process? Can you raise your hands? How many of you know what is Haber-Bosch process? That's very unfortunate. Haber-Bosch process is the process for making ammonia. The chemical fertilizer urea is used by this process where the atmospheric nitrogen is converted into ammonia or eventually to urea at very high temperature. And for this high temperature, you require very high fuel consumption to reach to that high level. And globally, the urea is being produced by this process, and it was given a Nobel Prize at one point of time because that gave the hope that the hunger of humanity can be met because of this nitrogen fertilizer through this Haber-Bosch process. But eventually we realized that this chemical fertilizer is creating havoc with the agriculture, with the soil. And therefore, we need to depend on biological nitrogen fixation for meeting the need of the crops. Because ultimately, if we continue to produce this urea fertilizer and give it at high subsidy to the farmers, this is at a very high cost of the climate because the global warming will continue to happen. So among the sustainable development goals, one of the sustainable development goals is to reduce the dependence on chemical fertilizers for crop productivity. And there, these nitrogen fixing bacteria are extremely important. For several years, people were not considering nitrogen fixation research on nitrogen fixing bacteria as important because this was not meeting the expectations. But in the light of current global warming, now we know that we have to go back and find out the bacterial strains which can support the growth of the non-legume crops. Well, with this background, I will try to talk about my research which is focused on Azospirillum brasilense. And if you look at this root, this is plant root, and this region around is called rhizosphere. Rhizosphere is the region around the roots which is influenced by the root activity, and particularly the root exudates, because after the photosynthesis, a huge amount of these photosynthate is released. It is released. So this is released into the by the roots, and this photosynthate becomes a source of growth and multiplication by the rhizosphere bacteria. So the rhizosphere is the zone of influence by the root activity. And because the roots are releasing a lot of root exudates, a lot of photosynthetes, so bacteria need to multiply, and for that they try to preferably move towards the roots in order to get their carbon source, in order to meet their carbon requirements, energy requirements. And therefore, rhizosphere is a very intense zone of competition. Large number of bacteria are present there. They are fighting with each other. Fast mobility, producing some antibiotics, other trying to protect itself from antibiotics. So there is a great arms race which is going on in the rhizosphere. And the winner is eventually able to colonize the rice root. The root itself doesn't like alien bacteria to colonize. It produces oxidative, this phenomena called oxidative burst. Because of that, a lot of reactive oxygen species, they are released by the plant roots to kill bacteria. So the bacteria which are able to sustain all this onslaught from the plant, from other bacteria, they survive, and then they start utilizing carbon source, which is released by the plant, and then they start multiplying there, whether pathogens or beneficial. Now, carbon source is the very important criteria. All this fight is for the carbon source. So, the ability to utilize carbon source is extremely critical, extremely important for the bacterium in order to colonize the rice roots, besides many other traits which are described here. For example, acquisition of and solubilization of nutrients, that is through siderophores for iron nutrition, phosphate solubilization, nitrification and nitrogen fixation. Antioxidant activities are, as I mentioned, many reactive oxygen species are produced by the roots as well as many other bacteria, and the rhizosphere bacterium needs these enzymes in order to detoxify reactive oxygen species. Biofilm, for ability to form biofilm, that the bacteria, once they multiply, they should form a layer over the roots so that they are in constant touch and they can absorb the nutrients which are released. And then volatile organic compounds, they have specific role in the ecology. Antimicrobial compounds, and I mentioned to you that there are many antimicrobial compounds, antibiotics which are released by some bacteria. So the other bacteria need to defend themselves from these kind of compounds. Motility and chemotaxis, these are extremely important because if the plant is releasing some compound, this is of use for X bacterium, and if this bacterium is not able to sense that its useful compound is being released by the plant, it will not move towards it. So this chemotaxis, to sense what is being released by the plant and then have the wherewithal, that means the flagella, so as to move towards the plant roots, that is extremely important in order to be a very rhizocompetent bacterium. So there are many factors for a bacterium to be rhizocompetent, and that is what this slide shows. So for highly competitive rhizobacterium, these properties in a bacterium need to be there. Now, Azospirillum is one of the most popular plant growth promoting rhizobacterium, and there are many formulations across the world. They are being used around the world for promoting growth of many crop plants, but most probably many millets as well as cereals. Now, millets are becoming important, and therefore, for many of the millets, Azospirillum is extremely important bioinoculant. There was one slide which I'm not able to show. And this slide, I don't know why it's not coming. This slide shows that what is the main mechanism of Azospirillum for plant growth promotion. The Azospirillum releases the phytohormone indole acid, and so do many pathogenic bacteria. They also release the phytohormones. Because of this, the root system proliferates. You have large number of root hairs, and the secondary roots are many. Because of that, the root surface area increases, and because of this increase in the root surface area, the ability of the plant to assimilate more water and nutrition, that increases. And therefore, this Azospirillum is very effective biofertilizer in the areas where there is a lack of water, drought affected areas. So there, the water availability is less, so this bacterium is able to increase the surface area of the plant root, so it absorbs more water and more nutrients, and therefore it can promote the plant growth. That is the main mechanism this bacterium uses for plant growth promotion. Now, so far, there are about 25 species of Azospirillum which have been reported. But the the oldest species which were described by Johanna Dobriner are Azospirillum lipoferum and Azospirillum brasilense. Both of them were initially described from Brazil, But in my lab, students have isolated large number of different Azospirillum, brasilense, lycophyrum strains. But these are the original descriptions. Now, if you look at their carbon source utilization capability, I'm sorry the projection is a little small, but I would like to read here that there are the characteristics about utilization of different carbon compounds. This is also not working well. But anyway, so you look at these the red rectangles. You can see the fructose, which is this is not fructose is being utilized by lipofilament and brasilense both. Glucose is utilized by lipofilament, not by brasilense. Glycerol is utilized by both brasilense as well as lipofilament. And dicarboxylates, which is not mentioned here, dicarboxylates, that is malate, succinate, and fumarate, these are the preferred sources of carbon for Azospirillum brasilense. And I will show some of the studies which we have undertaken about the mechanism of the uptake of dicarboxylates and fructose and glycerol and glucose. These four compounds we have focused, and we will share that how we have tried to address that how important they are for their growth, behavior, and colonization and plant growth promotion. Now here is a simple slide. I mean, some of the students may find it slightly difficult, but I will try to explain to you. You see, this is on top left, there is a growth curve. So you see that there is four curves are there. The one at the bottom shows the growth on fructose, which is very slow. Whereas on malate, succinate, and fumarate, the top three, it is growing very fast and it is reaching to high yield, high growth yield in, let's say, 24 hours, or when you look at the growth curve. Now, when you compare the growth on the plate, you can see that the malate, succinate, and fumarate, these are the three, where it is growing very fast. This is a semi-solid medium it is growing, because when you put bacterium in the center, this bacterium starts using the carbon compounds, and when this is finished, it moves to the periphery and further and further. And that is how this zone, looking at this, you can feel that this is utilizing carbon source very fast. Whereas you look at the fructose, it remains at the center and grows very slowly. So fructose is a poor source of carbon, whereas dicarboxylates are good sources of carbon. And when you do the STS page, a method to identify the proteins, a simple method, and you compare, you find that there is this protein. If you compare this protein profile, you find that there is one protein, and this protein is present in malate and to some extent in succinate and fumarate, but not in fructose. We picked out this protein, you cut out from the gel and identify. There are several methods of identification. Easier method is based on Marley-Toff. And by Marley-Toff, it is possible to identify what is this protein. And when we identified it, it is called a protein which is called DCTP. Now, what does DCTP do? There is a dicarboxylate transport system which consists of three proteins. This is DCTPQM on the left. Do we have any pointer which I can use, because this is not working? So you can see here that on the membrane, you can see that there are two proteins which are integral membrane proteins, DCTQ and DCTM, and there is a protein above which is called DCTP. This is a periplasmic protein. Periplasmic means a protein present between inner membrane and outer membrane. So this protein is a dicarboxylate binding protein. It binds to the dicarboxylate, malate, succinate or fumarate, and then it brings it inside. So this is able to bring it inside. Now if DCTP is not there, then the dicarboxylate cannot be taken in. So this is what we try to see. What we did was that after identifying this protein, DCTP, we went to the genome, found out the gene, and we inactivated this gene by inserting an antibiotic resistance gene cassette. When you inactivate it, then DCTP is supposed to have gone. And whether it has gone or not, that can be verified by SDS page again. You can see this. Sorry. You can see this here. This DCTP protein is present in the wild type, but not in the mutant. So the protein is gone, and because of this, you can see the growth also, where on the plate above, the wild type SP7 makes a wider ring, whereas the mutant is able to grow very slowly. So that means the dicarboxylate transport is affected, adversely affected. So you can see the organization of this gene, DCTP-QM, this is a kind of operon, and DCTP is the first gene, Q and M, they are the integral membrane proteins. And so we have shown that the dicarboxylate transport, see, so this is similarly shown in the growth curve, and dicarboxylate transport protein is important for uptake of dicarboxylate. That's what we established by this. Now, there is another dicarboxylate transport system, because you have seen in the previous slide that the mutant is not completely able to take up dicarboxylate. It is still able to take up some dicarboxylate. That means there should be some alternative mechanism to take dicarboxylate. And when we looked into the genome, we found another system which is called this DCT-ABD. DCT-A is a transporter, whereas DCT-D is a transcriptional activator. This is a membrane protein. So DCT-ABD system, we inactivated DCT-A to see that if we inactivate DCT-A, what will be the effect on the transport. And then we found that it does affect the transport, but not as much as DCTP. Now, okay. So here, what I have done is that I have taken these mutants. This is a wild type. This is DCT-A mutant. This is DCTP mutant. And this is the double mutant. These are the strains which are tagged with green fluorescent protein. Because if you inoculate bacterium into the plant root, you will not be able to see it under microscope. In order to make it visible, you tag it with the green fluorescent protein, and when you give ultraviolet light, then this green fluorescent protein fluoresces. And wherever green fluorescence is there, you can feel that the bacterium is there. Now, if you look at the SP7, it is able to colonize the root of a foxtail millet. This is a plant called foxtail millet. This is a C4 plant, and in the foxtail millet, this colonizes very well. If you inoculate this DCT-A mutant, which is slowly reducing the dicarboxylate transport, the colonization is not reduced substantially. It is reduced, but not that much. Or if you take DCTP mutant and GFP, green fluorescent protein tagged strain, if you colonize, you find that this is not present. This is very few colonization is there. Or if you take double mutant, you find this not there at all. So that clearly shows that DCTP protein is extremely important for dicarboxylate transport. And DCT-A is not that important. So dicarboxylate transport is important for the bacterium to take up this dicarboxylates. Now, whether the dicarboxylate transport is important for the growth of the plant, for this we have done some experiments about colonization, and we find that this DCTP mutant, the root length, root hairs, as well as secondary root number is very low, whereas DCT-A and wild type, this colonization shows that root is growing normally. But when you are inoculating with the DCTP mutant and double mutant, the root proliferation is not as much. So this is early stage, and later stage you can see these difference much more prominently. So this further establishes that the ability of utilizing dicarboxylate affects the plant colonization as well as plant growth promotion. So that is one thing which we were able to establish. Now the second component. The second component is that one major difference between Azospirillum lipophyrum and brasilense is that brasilense cannot utilize glucose, whereas lipophorum can utilize glucose. Now we wanted to show that this inability of Azospirillum brasilense to colonize rice is because of the lack of its ability to utilize glucose is based on this, that brasilense is a poor colonizer of rice, whereas lipophorum can do that. The reason is that you see this root exudate analysis. From the roots, the exudates were analyzed at 7, 14, 21, 28 days, and you find that the glucose is the maximum. In the early stages of the root exudation, you find that maximum amount of glucose is there, which gradually declines. So for initial colonization, if the bacterium is having ability to utilize glucose, it will be able to colonize well. But brasilense doesn't have this ability, and therefore brasilense is a poor colonizer of rice. We wanted brasilense to become a potent colonizer of rice roots, and for that we thought that if we introduce the ability to utilize glucose, whether this will be able to colonize well and promote rice growth or not. So for this we did a few experiments. Now, how is glucose transported? See how we conceptualize, how we made the hypothesis. The glucose transport in most of the bacteria happens by a phosphotransferase system. And in this phosphotransferase system, there is a phosphoenol pyruvate. From this, phosphate is transferred to a protein called E1, then to HPR, then to E2A, and then it goes to E2BC complex. Now in E2C is the membrane protein, and when the glucose from outside comes, at this level, this is phosphorylated, and phosphorylated glucose can go only inside, it cannot go outside. So that is the mechanism to make sure that glucose remains inside after its transport, because it's phosphorylated. So this is the glucose phosphotransferase system which is present in many bacteria, not in brasilense, but present in the lipophorum, which is its cousin. So you can see here that this is a situation of lipophorum, and in the lipophorum you have this phosphoenol pyruvate, E1, HPR, E2A, and E2BC protein, E2B protein, E2C protein is there, and it also has an enzyme called glucose 6-phosphate dehydrogenase. This in red, these proteins are absent in brasilense. Can we pick up these genes from lipophorum and transfer in brasilense and see whether brasilense can utilize glucose? This was our hypothesis, and that is where we started working. When we looked at the genes which are responsible for glucose phosphotransferase system, we found there were some other genes like this NAG-A and GFPT, this is N-acetylglucosamine transglycosylase. These two genes were present along with the E2BC genes and E2A. So we did several permutations and combinations, that whether this gene alone, glucose 6-phosphate dehydrogenase alone, can confer the ability to utilize glucose. Can these two genes, NAG-A and GFPT, confer the ability to utilize glucose? Likewise, we did several permutations and combinations, and we found out that G4, which consists of NAG-A, GFPT, E2BC, and E2A, when you provide this, then Azospirillum brasilense is able to utilize glucose. But if you provide this G4 plus G, that is the glucose 6-phosphate dehydrogenase, it grows much better. This is what is shown here, G plus G4, that is G plus G4. If you provide this combination, it is able to utilize glucose much better. So our next step, this is the, sorry. Yeah, so here what we did, that these 4 genes plus glucose 6-phosphate dehydrogenase, we put it together under one transcriptional unit under the control of kanamycin promoter, and we transferred it into the SP7 wild type. And you can see that SP7, which is without this cassette, is not able to grow on glucose, but here it is growing very well. That means it is able to utilize glucose if you transfer these 5 genes under one promoter. Now, it is growing actually better than Azospirillum lipoferum, from which these genes have been taken. Lipoferum is the source. From this, if you transfer into brasilense, which was not utilizing glucose, now Azospirillum brasilense is able to grow much better than even its parent lipoferum, from where the genes have been taken. And we have shown it that here, that after 96 hours of growth, you find that engineered strain is able to utilize this glucose much better than lipoferum, where the glucose level is still high. So it is very efficient in glucose utilization. So we proved that this engineered strain is able to utilize glucose much better than its parent, as well as Azospirillum lipoferum, from where the genes were taken. We wanted to see whether this strain of Azospirillum brasilense can colonize the rice or not, and then whether it can promote the plant growth or not. And for that, we did the next experiment, and this experiment was similar to the dicarboxylate. Here, you can see this is the wild type SP7, and this SP7 engineered with the G5 cassette, where all the 4 genes plus glucose 6-phosphate dehydrogenase, all of them are engineered. And then here is the dicarboxylate transport mutant, which I described earlier, and this is the mutant which was having this G5 cassette. So now you can see that the wild type SP7, it was able to colonize rice because this can utilize some other carbon sources which are present in the rice rhizosphere. But when you insert this cassette, G5 cassette, you can see much better, very profuse green fluorescence. That means its colonization ability has increased considerably. But if you look at the dicarboxylate transport mutant, so the growth here, SP7 colonization here is because of its ability to colonize, to utilize dicarboxylate. But if you make it handicapped in utilizing dicarboxylate, that is by creating this DCTP mutant, where you can see very little colonization is there. But here, if you transfer this G5 cassette, you can see the colonization ability improves. So that clearly shows that the ability to colonize rice improves because of these 5 genes which have been taken from Azospirillum lipoferum. Can it promote plant growth? It has improved the colonization, but can it improve the plant growth? That experiment was done, and you can see that this is the outcome. I would like to draw your attention to this and this. This is colonized by wild type, and this is colonized by wild type plus the cassette, which has got all the 5 genes. And you can see the difference in the biomass, that this is colonized by wild type, and this is inoculated by no bacteria, no additional bacteria. So you can see this one, this shows plant growth promotion by colonization of SP7, inoculation of SP7. But engineered strain shows much better growth of the rice in the pot system. And here is the dicarboxylate transport mutant, but this mutant, when it is engineered, it shows much better growth. So this also now clearly shows that engineering Azospirillum brasilense with glucose utilization cassette is not only improves the colonization in the rice root, but also improves the plant growth promotion. Now moving ahead, I think I will just go to a couple of more aspects. That Azospirillum brasilense as such does not utilize ethanol, alcohol. It is having adverse effect on the growth of Azospirillum brasilense. Now, we have done the proteomic analysis, and in the proteomic analysis we found that if you give glycerol, you find that these genes, this protein, protein alcohol dehydrogenase, another aldehyde dehydrogenase, and another aldehyde ethanol dehydrogenase, these enzymes are upregulated. What do these enzymes upregulation means? That means they can probably improve the ethanol utilization because these are alcohol dehydrogenases and aldehyde dehydrogenases. So we did some experiments further and found out that this fructose and glycerol, these two sources, these compounds can increase the uptake of the alcohol dehydrogenase. Now, when you do this experiment, you find that in the mallate medium, if you provide mallate as a carbon source, the growth of Azospirillum brasilense is inhibited by ethanol. But if you grow it in fructose or glycerol, actually the growth is promoted. This is the wild type, and this is the wild type supplemented with ethanol. But here, this is the wild type, and this is the wild type supplemented with ethanol. So what the conclusion is that in presence of mallate medium, ethanol is toxic, ethanol is not utilized. But in presence of fructose and glycerol, this is utilized because fructose and glycerol are able to induce the genes or the proteins or the enzymes which are responsible for assimilation of ethanol. So this is kind of this phenomena is called co-metabolism. You give a single carbon compound, nothing happens. Actually, this may have toxic effect. But if you give it along with another carbon compound, then this second carbon compound is able to induce the proteins which are utilized, which are required for utilization of the alcohol. So this is called co-metabolism, and that is the phenomena which we were able to see here. I will not go into the detail. Only thing is that here, that normally ethanol is converted into acetaldehyde, and acetaldehyde is converted into acetate, and this goes into glyoxylate cycle by these two proteins, XA and aldehyde A. And when mallate is present, mallate inhibits the production of XA and ALDA so that ethanol is not converted into ethanol. But in presence of glycerol and fructose, these two proteins are produced, and because of that, ethanol is converted into acetate, which is utilized as a carbon source. So this is the mechanism which we were able to find out, that why in presence of fructose and glycerol this is utilized, ethanol is utilized, but in presence of mallate it is not utilized. Now I'll take the last aspect, which is very interesting. We saw fructose is utilized by both brasilense as well as lipofenon. We wanted to see what are the proteins which are upregulated by fructose in Azospirillum brasilense. When we analyzed, we found 156 proteins which were upregulated. But there were a few proteins which were of considerable interest. And these proteins are shown here. They all are component of a type 6 secretion system. What is type 6 secretion system? This is a complex of proteins which are able to inject toxins into other bacteria or other fungi in order to kill them. So this is a great armory in the bacteria which are having this type 6 secretion system. So this type 6 secretion system proteins were upregulated besides many other proteins which were upregulated. But I just want to draw your attention to only these proteins which are part of a kind of a system, and I will show you that how this is. So this was an evidence that type 6 secretion system may be functional. Whether it is functional or not, that is what we were supposed to show. So we did some more experiments to show that this fructose is actually taken up by. So fructose is taken up. There are two types of fructose, phosphotransferase systems, and it's only one of them which is utilized for fructose uptake. Through A1, when you knock out, the fructose uptake is completely gone. But through A2, this is the second one. If you knock it out, there is no effect. That means through A1, one operon, this is utilized for the fructose uptake. So that's what we have shown. And now here again, you see that these are the proteins. This is type 6 secretion system proteins. You can see number of these proteins. These are part of the operon. These are upregulated. This is how they are organized in the genome. So we wanted to show whether in reality Azospirillum can kill other bacteria or not. So this inhibition is a contact dependent inhibition. So the two bacteria come in contact and they make an injection-like structure, and through this injection, the toxin is injected into the other bacterium so that it is killed. Now, we have found that this is induced by fructose. So in presence of fructose, this system should work. In the absence of fructose, this system should not work. And this is what we have shown in the next slide. You can see that this is how this system works. So this is complete system which is consisting of many type 6 secretion system, and when it comes in contact with the bacterium, this is a kind of pumping, and it pumps these proteins which go into the neighboring bacterium and is able to kill that. So this is complete very nice complex, like you have a gun or a 303 and you shoot it and you fire. So similar kind of phenomena is taking place here, that it is pulled and then fired, and this kind of structure is present, which is already shown in several other bacteria, but we have shown it for the first time in Azospirillum. That presence of this confers competitive advantage to Azospirillum brasilense in being a most competent rhizosphere bacteria, because it is able to kill many other bacteria. Now the evidence, you can see this, that this is the E. coli. These blue spots are the colonies of E. coli which are expressing beta-galactosidase activity. Because of that you see them blue. And there is a lawn of Azospirillum brasilense. This lawn is there. You see, so this is Luria broth. In the Luria broth, these colonies of E. coli are growing well, and whether it is sp7 bacterium or not, they are growing equally well. But you can see that in the fructose, this sp7 is able to inhibit the growth of the E. coli. This plate is nearly clean, and that shows that sp7 in presence of fructose, the brasilense in presence of fructose is able to kill this E. coli, but in presence of Luria broth, in the absence of fructose, it is not able to kill. So that clearly shows that killing is taking place, and this killing related proteins are upregulated because of fructose. Now the next step was that whether the type 6 secretion system was responsible for this killing or not. So what we did was, we inactivated some of the genes which are essential component of the type 6 secretion system, and tried to see whether in the presence of fructose, this bacterium is still able to kill the E. coli or not. So in the next slide you see that this is the E. coli, and this is the agrobacterium. Agrobacterium is another rhizobacterium which is killed by this sp7. You can see this is a clean plate. This is growing here, but in presence of sp7 in fructose, this is being killed. But the next step is important, that even after the presence of fructose, this mutant of sp7, where these genes are inactivated, it is able to allow the growth of this, not that much, but still it is able to grow. So that means that in the absence of the genes required for making type secretion system, the inhibition becomes less effective as compared to the wild type strain, where the type 6 secretion system is intact. So the type 6 secretion system is actually involved in killing of E. coli as well as agrobacterium. So we have given two examples to show that this is able to kill. But you see, if I give you previous example, this is a plate here, a small picture. So this was the lawn of Azospirillum lipophorum, and in the center we kept a colony of Azospirillum brasilense. Now Azospirillum brasilense was able to kill its cousin Azospirillum lipophorum. We have not done further experiments on that, but Azospirillum brasilense, if you look at brasilense and lipophorum, brasilense is considered the most popular and most effective PGPR. Lipophorum is not. The reason is that lipophorum survives less in the rhizosphere, but brasilense can survive better because this has the ability to kill the neighboring bacteria. So this is the evidence which we have provided, that E. coli and agrobacterium, both of them can be killed by Azospirillum brasilense because of the type 6 secretion system. Now these are the highlights. I will not go into the details of it. I have already explained to you all the details. And these are the two students, Vijay Shankar Singh, who initiated this work on the dicarboxylate and glucose, and later on Sushant joined this Vijay in carrying out further studies, and this our paper on the fructose utilization and fructose induced type 6 secretion system. This paper is under revision for applied and neutral microbiology. I hope that in a couple of months it should be online. So this is a very nice paper. The editor got very excited looking at this data. So this is yet another evidence that brasilense is equipped with many traits to make it a very successful rhizocompetent bacteria. So with this, I would like to thank you all for your kind attention, and to the organizers for giving me the honor, and all of you for your patience in listening to me. Now, if you have any question, I would like to address them.
ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರಿಗೆ ಏನಾದರೂ ಪ್ರಶ್ನೆಗಳಿದ್ದರೆ ದಯಮಾಡಿ ಕೈ ಎತ್ತಿ, ಮೈಕ್ನ ನಿಮ್ಮ ಬಳಿಗೆ ನಾವು ಪಾಸ್ ಮಾಡ್ತೀವಿ. ನಿಮ್ಮನ್ನು ಪರಿಚಯಿಸಿಕೊಂಡ ನಂತರ ಪ್ರಶ್ನೆನ ಚುಟುಕಾಗಿ ದಯಮಾಡಿ ಸಂಪನ್ಮೂಲಗಳಿಗೆ ಕೇಳ್ಬೋದು. ಯಾವುದಾದ್ರು ಅನುಮಾನಗಳಿದ್ದರೆ ದಯಮಾಡಿ ಪರಿಹರಿಸ್ಕೋಬೋದು.
So the student should not feel any hesitation, because I am a teacher. So look at me from an angle of teacher, not only as a scientist. So I'm supposed to address to your queries. So feel free to ask questions. So this clearly shows that you have not understood anything.
I hope they have understood everything.
Small question, sir. So, sir, you said like it is a type 6 secretion system inhibiting or controlling E. coli. So, is there any studies that have been carried out from your group? Like it is, how does it induces a plant defense system, especially in millets, for controlling any mycotoxin producing fungi, or it is colonization of any Aspergillus or Fusarium species? Any relation that has been established, sir? Maybe that could have been helpful for us also.
See, we are working on Azospirillum.
Yes, sir.
But I am aware of the work of Gopalji Jha at NIPGR, who is working on Burkholderia. And Burkholderia has a type 6 secretion system, and this Burkholderia is able to kill several fungi, pathogenic fungi. So his work has received a lot of limelight because the bacterial strain which is able to kill pathogenic fungi, that is what he has demonstrated. We have not done anything so far on Azospirillum. We have only worked on 2 bacteria, but maybe in the next step we would like to work on killing off the fungi.
Thank you, sir.
Thank you, Professor Anil Kumar Tripathi. your deep insights and scholarly discourse have enriched us all. We are truly grateful for sharing your knowledge and expertise with us on this occasion. Thank you, sir. As we draw curtains on this memorable event, it's our duty to extend heartfelt gratitude to all those who have contributed in making today's endowment lecture a resounding success. Now, I have the honor to invite a senior academician, Professor Ines Raju, who also serves as the chairperson of the Department of Environmental Science, to propose vote of thanks.
Everyone, namaskara. Good afternoon. It is my privilege to propose a vote of thanks at the end of this endowment lecture on Paul Neergaard and Professor Viggo Smedegaard endowment lecture series. First of all, I would like to thank our Honorable Vice Chancellor who has presided over this function and accepting our invitation to preside this function and make this function very successfully in his absence. And also I thank my mentor, Professor S.R. Niranjana, who is the chief guest of today's endowment lecture, and also the Vice Chairman of the Karnataka State Council for Higher Education, for accepting our invitation and make his presence in this occasion. I also thank our Honorable Registrar Shailaja Madam for constant support to conduct this endowment lecture. On behalf of the committee, and I thank Shailaja Madam for the presence in this function. And also I thank today's speaker on the today's speaker, Professor Tripathi from Banaras Hindu University, Dr. Anil Kumar Tripathi from Banaras Hindu University, who has enlightened us by giving a wonderful lecture on the carbon source utilization ability for Rhizocompetence, the PGPR, using Azospirillum brasilensis, which is an eco-friendly approach for this to save the earth from the use of chemical fertilizers. So on behalf of the committee and the University of Mysore, we would like to extend my heartfelt thanks to Professor Tripathi sir. And also thank Dr. Amrutesh, who is the convener of this function, endowment lecture, who is successfully convening the programs for the last many days. The last week also he has conducted the same type of endowment lecture, inviting eminent scholars from the country. So on behalf of the University of Mysore, I would like to thank Dr. Amrutesh. I also thank Professor Prakash sir is here, and also the professor from Silviculture Department, Professor Subramanian, and also the research scholars from various departments, faculty members, students, media persons, and all the participants for your presence and patience hearing. And also thank Dr. Sudesh from the Central University of Kerala, Department of Environmental Science, for his presence. And also thank Dr. Loit, who is one of the member of the program, the endowment lecture program, for his comparing and his support. So finally, once again, I thank one and all for the success of this function. Thank you very much.
ಈಗ ನಮ್ಮ ರಾಷ್ಟ್ರಗೀತೆಯ ಧ್ವನಿ ಮುದ್ರಿಕೆಯನ್ನು ತಮ್ಮ ಮುಂದೆ ಬಿತ್ತರಿಸಲಾಗುವುದು. ರಾಷ್ಟ್ರಗೀತೆಗೆ ಗೌರವ ಸೂಚಕವಾಗಿ ದಯಮಾಡಿ ಎಲ್ಲರೂ ಇದು ನಿಲ್ಲಬೇಕು.
अधिनायक जय हे भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा, विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा, तव शुभ नामे जागे, तव शुभ आशीष मांगे, गाहे तव जय गाथा, जन गण मंगल मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
ಬಂಧುಗಳೇ, ಸಭಾಂಗಣದ ಹೊರಗಡೆ ತಮ್ಮೆಲ್ಲರಿಗೂ ಕೂಡ ಭೋಜನದ ವ್ಯವಸ್ಥೆ ಮಾಡಲಾಗಿದೆ. ತಾವೆಲ್ಲರೂ ಭೋಜನ ಸ್ವೀಕರಿಸಿಯೇ ತೆರಳಬೇಕಾಗಿ ಆಯೋಜಕರ ಪರವಾಗಿ ವಿನಂತಿಸ್ತೇವೆ.